Hi everyone. Today we're going to cover chapter 6. And chapter 6 is the last chapter in our discussion of macromolecules. So to date, we have previously talked about 3 different groups of macromolecules, right? We talked about proteins. We have talked about carbs. And we've talked about nucleic acids, right? And all 3 of these were macromolecules. But in addition to being macromolecules, they were also polymers. So they were made up of individual repeating units, right? Lipids are also macromolecules, but they are not necessarily polymers. There are some that can be, but in general, they do not have to be polymers. So these are going to be a little different than some of the molecules we previously talked about. Another thing that's going to be a little bit different about this chapter is we're going to move away from our discussion of just lipids and start talking about lipids in the context of cell membranes and how cell membranes work. So this is a fairly long and pretty important chapter, so I'm going to be breaking it up into 2 parts for you, and both parts will be on the exam, okay? So let's take a look at what you should be getting out of this chapter. First of all, we're going to talk a bit about the structure and function of lipids. We're going to discover that they will form bilayers, and lipid bilayers are going to be the basis for cell membranes. And then we're going to hone in on these lipid bilayers to learn how we can move things across lipid bilayers. And this is going to lay the foundation for our conversation about how we get things into and out of cells. So we'll be talking about diffusion and osmosis, and we'll also be talking about how you can insert proteins into lipid bilayers to transport things across those membranes, okay? Now let's think for a second about the invention of the lipid bilayer. So I just told you that lipid bilayers are cell membranes, and I'm going to show you some diagrams of that in a minute. But right now I want you to think back to like a primordial Earth when everything was happening in water. And we learned in chapter 1, I believe, that there was an experiment that showed us in early Earth when we had conditions where we had water and we had things like lightning. So I'm going to try to draw a terrible lightning bolt here, right? And here's a thundercloud. Okay, so we had an energy input and we had some simple molecules. We learned in chapter 1 that you could end up with some other larger macromolecules. We also learned in chapter, I think 4, when we talked about nucleic acids, that RNA could actually function as an enzyme to make itself, so it can catalyze its own synthesis. Today, we're going to learn that lipids can spontaneously form bubbles, so to speak, these lipid membranes that are made of 2 layers of lipids. So think of this as being inside some water. Now we have a bubble that's made up of oil. That's probably the easiest way to think of it. So you have water out here, and you have water in here. And this is your membrane. So this is a hallmark achievement in the evolution of life, because what we've done now by creating this little micro environment inside this lipid membrane, what we're able to do is any chemical reactants that we've gotten trapped in here, let's say that we got some nucleotides and a ribozyme, which remember is RNA, that is capable of catalyzing a chemical reaction, some of which can catalyze their own synthesis. So let's just say we had some ribonucleotides and a ribozyme in here, and we have them all kind of clustered together inside this membrane. We are increasing the likelihood that they will react with one another. So having a cell membrane is going to increase the rate of chemical reactions, because things aren't just floating about in this large puddle of water, right? So it's going to increase the rate of reactions. And I'm going to abbreviate reactions R, X, and S. The second thing that this bubble of oil is going to do, oil doesn't dissolve in water very well, does it? So it's going to create a barrier. Some things will be able to get across this barrier while other things will not. So we say that these membranes are selectively permeable. And so we are going to talk a lot about how we get things into and out of the cell. And that's important. You don't want everything in the cell floating out, and you don't want everything in the outer environment getting in. Okay? So these are kind of the big important things about having a cell membrane that's composed of a lipid bilayer. But let's go ahead and look more closely at lipids themselves. All right, so let's just talk about their structure first and foremost. All right? So one of the first things that I want you to notice is that what we see is a lot of carbon, 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 and a lot of hydrogen, okay? And when you see this here, I want you to think of this as being a carbon with attached to a carbon here, attached to a carbon here. And when you see that H2, like this right here, just understand that that really is shorthand for a carbon bond on either side, sorry, a hydrogen bonded to either side, okay? So lots of carbons, lots of hydrogens, okay? So in general, we say that lipids are hydrocarbons because that's literally the majority of what they're made of. Oops. Now, one of the defining characteristics of lipids is that they are not water soluble. They are, in fact, what we call hydrophobic. What does that mean? Well, let's break that word down into its component parts. Hydro, of course, referring to water, sometimes hydrogen, but water. Phobic, meaning afraid. So water afraid, that should tell you that what this really means is not water soluble. And you guys know that. You know that lipids are not water soluble. If you're making, say, if you have a bottle of Italian salad dressing, you know that the oil and the water separate with the oil on top and the water on the bottom. And if you shake it up, you can get little bubbles of oil kind of suspended in that water, but in general, they don't mix very well. Now, why is it that these are not water soluble? Well, remember that for things to be water soluble, they have to be polar. For things to be polar, there has to be an electronegative atom like carbon present. So in this molecule here, we don't see, an electronegative atom like oxygen present. I misspoke. So remember how oxygen likes to hog electrons? It likes to keep them to itself. And when it does that, it creates a slight negative charge, right? It also, by virtue of pulling all the electrons toward itself, makes this area here slightly positive because it's taking away their electrons. So that's how polarity is created, by the presence of an electronegative atom like oxygen. Carbon, you see we have some equivalent bonds on all sides. Carbon isn't really tugging at anyone else's electrons. Hydrogen is sharing evenly. So we don't have any partial positive or partial negative charges down here, do we? So that is going to create a situation where this region of this molecule is nonpolar, right? Now you may be saying to yourself, I'm showing you 3 different examples of lipids, of chemical structure of lipids. These are 3 different ones. This one's called an isoprenoid. This one's called a fatty acid. And I'll talk about the difference between saturated and unsaturated in just a second. But in this fatty acid, you're like, well, okay, this region's nonpolar, but this region up here is polar. So why isn't this water soluble? It just has to do with the proportion of this molecule that is nonpolar. So this region down here, all of this down here is nonpolar, isn't it? And if you look at the size of the nonpolar region relative to the size of the polar region, the nonpolar region definitely wins out for size. So this is going to be an overwhelmingly nonpolar molecule. Now, one thing that I wanted to point out, I told you that lipids are not really polymers, or they don't have to be polymers. So let's take a look at our lipids that I have as examples for you here. And let's start with this one. So this is what's called an isoprenoid. An isoprenoid is made up of isoprene units. So this one is kind of a polymer. Can you see that we have repeating units here? Okay, that is a polymer, right? But then let's look at these. These are not made up of repeating units. You've got a carboxyl group here, and then it's just a bunch of hydrogens and carbons. Here you have a carboxyl group, hydrogens and carbons, carbon-carbon double bond, hydrogen and carbons. So in general, you may have some lipids that are polymers, but they don't have to be, okay? Now let's talk a little bit more about, let me get rid of this, a little bit more about the structure of lipids, and let's hone in on a concept called bond saturation, okay? So, in thinking about bond saturation, this is a good example for you on this side where we're looking at these two fatty acids, okay? This is what we call a saturated fatty acid. Now, what does that mean? Let's think about the term saturated. If you had a sponge and you submerged it in water and you let it soak up as much water as it could completely hold until it can't hold any more water, then you would say that that sponge was saturated with water because it had as much as it could without being able to hold any more. Well, let's kind of look at this. Let's take a look at this carbon right here. I'm going to just hone in on this carbon. I'm going to blow it up, make a little drawing. If we look at this carbon right here, make that our central carbon, what you really see is you've got a carbon bound to another carbon here, a carbon bound to another carbon here, and then I've got an H2. So on either side, I have a hydrogen, right? Okay, excellent. Now, remember from our previous conversations that carbon has a maximum of 4 bonds that it can make to fulfill its outer valence shell. This has as many bonds as it can possibly participate in. And so therefore, this is a saturated carbon. And if you look, each of these carbons has 4 bonds. So each of these carbons is what we call saturated. Now, let's take a look at this carbon in contrast, okay? So I'm going to draw this carbon here. And then we see that it's got a double bond to another carbon. It's got a single bond to this carbon. And then it's also bound to a hydrogen. All right. Now it's still got 4 bonds, but it's only bound to 3 other atoms, right? So these two are sharing electrons. It's got a double bond, but only 3 atoms. It's not saturated. It's not participating with the maximum number of atoms that it could possibly interact with. So it's an unsaturated carbon atom, okay? So why is this important? Well, it turns out whether a lipid is saturated or unsaturated is important to whether or not you're looking at a lipid that is a liquid or a solid. Okay, what do I mean by that? Well, let's look at this next slide. All right, so butter is a very delicious solid fat, isn't it? Shortening is a delicious solid fat. So what happens with butter is it's got a fatty acid, so this is the fatty acid end, and then what you can see here is this up and down. This is supposed to be a carbon, 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 right? Every point, remember me telling you in a previous lecture that we just understand that at each corner there's a carbon present. But notice there's no double bonds, okay? These are single bonds. And so what that means is that these lipids are able to stack on top of each other really tightly and form a solid like you see in butter, okay? Now let's switch over here to this oil situation. Here we have single carbon bonds, But look here, we've got a double bond, and then we come down and we've got a double bond. So this is a saturated situation, right? This is an unsaturated situation. So what happens with this unsaturated situation is it causes kinks. So if you were to try and lay these on top of each other, you really can't. They kind of stay kind of far apart from one another because they can't tightly pack like they can in butter. So they stay kind of far apart from one another and they remain in liquid form. So here we have a solid. And here we have a liquid, right? So one of the things that we're going to see as we move on to talk about cell membranes is the number of saturated lipids versus unsaturated lipids is going to contribute to how liquid our cell membranes are, or another way of saying that is the fluidity of cell membranes, okay? Now, another thing that determines sort of how solid a fat is how long these hydrocarbons are, how long these lipids are. So if we've got a bunch of short lipids stacked on one another, you get what I like to call a semi-solid like butter. So you've all experienced butter at room temperature where it's soft, but it's still holding together. But what about something like beeswax, okay? That's a nice rigid solid, even at room temperature. And the reason is we have these very long lipids that can lay on top of one another, okay? And so that contributes to how solid, or I guess the rigidity of a lipid. So two things that matter in terms of whether a lipid is a solid or a liquid and how solid it is. The first thing that matters is saturated versus unsaturated bonds. And the second thing that matters is how long the lipids are. Now, the reason that Crisco is pictured for you here is because it's undergone a process called hydrogenation. So Crisco is actually made from vegetable oil. You'll know that if you've ever used Crisco or your grandma's ever used Crisco to bake biscuits, it's called vegetable shortening, and that's because it comes from vegetable oil. So Crisco started life as something like this, but it underwent a process called hydrogenation where a chemist forced some hydrogens here and broke these carbon-carbon double bonds. So it took the vegetable oil, added hydrogens to break those double bonds so that you end up with something that looks like this, or looks like this, or looks like this, okay? So it's the process of hydrogenation takes unsaturated lipids and saturates them to make them solid. Now, one of the things that you may have heard is that polyunsaturated fats help protect against heart disease, while saturated fats are bad for your heart and they can contribute to like plaques inside your arteries. So you may have heard of atherosclerosis, which is a hardening of the arteries, and that's contributed to by saturated fats. We're not exactly sure why polyunsaturated fats, so poly meaning many, and unsaturated meaning double bonds. So if you're eating fats that are polyunsaturated, they've got lots of double bonds. We're not exactly sure why that protects against heart disease, but we know that it does, okay? Let's focus in on some particular types of lipids. So in this particular part of the lecture, I'm gonna focus in on types of lipids and their functions, okay? So one particular type of lipid that's going to be important in talking about cellular biology, they're called steroids, okay? Oh, sorry. They're called cholesterol. Steroids are made from cholesterol. I'm getting ahead of myself. Okay, cholesterol. You guys have heard of cholesterol, right? So cholesterol looks something like this right here, okay? It is an isoprenoid, you don't need to know that, but see, we don't have any oxygens except for right here. The majority of this molecule is nonpolar. We do have an oxygen up here, but most of it isn't gonna work, right? So cholesterol is important because it does many things. You can take cholesterol and you've got like a carbon-carbon double bond here. Cholesterol contributes to making cell membranes more rigid. So you can add it to membranes to make them more rigid. Okay. Another thing that cholesterol does is it's actually what we call the mother molecule for some other really, really important molecules in your body, primarily sex hormones and stress hormones. Oops. So if you've heard of testosterone and estrogen, those are steroids that are derived from cholesterol. If you've heard of cortisol, that is a steroid that is derived from cholesterol, and those are hormone types, okay? And then the last thing that cholesterol is involved in is it is involved in vitamin synthesis. Okay? So one function of lipids is we have a type of lipid called cholesterol that can be added to cell membranes to make them more solid. It can also be something that we use to make hormones with, and it can be something that we use to make vitamins with. What's another function of lipids and what's another type of lipid? Well, this type of lipid is called a triglyceride. So let's look at this. You may have heard of triglycerides before. All right, so what is a triglyceride? So I showed you in an earlier slide something called a free fatty acid or a fatty acid, where you have a carboxyl group up here, and then you have this long nonpolar chain. This right here is the same thing as this right here, okay? Now what we can do with this fatty acid is we can attach it to a molecule called glycerol. So this is glycerol up here, and we can actually use dehydration synthesis or a condensation reaction to attach one fatty acid here, one fatty acid here, and one fatty acid here. And when we do that, we end up with a glycerol with 1, 2, 3 fatty acids. This is a triglyceride. Now, you've probably heard of these. We measure these in people's bloodstreams, but the big deal about these is that they are used to form your body fat, your adipose tissue, okay? So when I say fat here, I'm talking about adipose. I'm talking about the fat that you have in your body. Now, what is the function of fat? Well, fats are important for energy storage, right? So that's another function. So we had our functions up here. Okay, these were functions of cholesterol. Here's another function, and it's a function of triglycerides. Okay, they are fats and they are involved in energy storage. Now, how is it that these triglycerides are good for energy storage? Let's take a look more closely at them on a molecular basis. So I have for you here a carbohydrate, and then I have for you here a fatty acid. Now let's just review how this is a carbohydrate and how this is a fatty acid. So here we have a carbon backbone, and remember that the formula for carbohydrate is CH2O, right? And then you can have however many of those units. So CH2O to the N. So CH2 and an O, CH2 and an O. So can you see that this is a carbohydrate here? Here we have a fatty acid because fatty acid or a lipid is a hydrocarbon. So we've got carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, okay? Hopefully you're with me so far. So this is a lipid, this is a carbohydrate. Now, you guys know that we often use sugars as fuel, and sugars are great fuel, right? Like glucose. I bet this is supposed to be glucose because it's got 1, 2, 3, 4, 5, 6 carbons. So this is probably meant to represent a glucose molecule. You know that glucose is great for energy. And so you can use this to fuel your body. But this actually has a greater energy storage capacity than this does. Now, why is that? It has to do with these bonds right here. If you look very closely, what you can see is that oxygen is pulling its old tricks, and it is hogging these electrons. It's holding these electrons close to itself, isn't it? So if you wanted to get the energy out of these chemical bonds, it's easier to snap this chemical bond off because these electrons are already almost completely snapped off than it is to snap this chemical bond apart because these are shared very evenly. So there's more energy stored here than there is stored here, okay? So let's think about that one more time. So we've got a carbon and an OH. I'm just gonna put the O. And the electrons are down here versus a carbon and a hydrogen, and the electrons are here. This is pretty easy to snap off. So it doesn't store that much energy, okay? It doesn't give off that much energy when we do the snapping because it's a pretty easy thing to do. This bond is longer. These things are more evenly shared. It's going to be a little bit harder to snap this guy off. So there's more energy stored in here. So that's why fats have a greater amount of energy stored than carbs do. As a matter of fact, if you were to look at 1 gram of carbohydrate, I believe that it is 4 calories versus, no, that's not right for gram. 4 calories of carbohydrate to 9 of fats, but I can't remember exactly what the unit is, okay? So this is less calories than this because this has more energy storage, okay? All right, let's look at another type of lipid. This third type of lipid is the one that we're gonna focus in on the most. This is a phospholipid. So let's look here. This should look familiar. This is a glycerol molecule, but here instead of having 3 fatty acid tails on it, we only have 2. In the third parking spot, instead of adding another fatty acid like we have with triglycerides, what we've done instead is we've stuck on a phosphate group. Now, phosphate groups, hopefully you can see, we have a negative charge here. And this is a fairly large part of the molecule. So because we have a negative charge, this part of the phospholipid is going to be soluble in water. But this part down here is insoluble in water. So we have a situation with these phospholipids where part of it can be dissolved in water while part of it cannot. When you have a molecule that is partially water soluble and partially insoluble, or partially hydrophilic and partially hydrophobic, the name for that is an amphipathic molecule. So amphi meaning both. Okay, and the big function of these guys is going to be that they form membranes. All right, so let's think a little bit about why that is. So if we look at these phospholipids with a phosphate head and then 2 fatty acid tails, what happens is if they're sitting in water, they're repelled by the water, but they tend to spontaneously form these bilipid circles like that, okay? Just like you see here. So we would have a whole bunch of little phosphate heads and lipid tails, okay? And then we'd have a bunch of phosphate heads and lipid tails. And so what you have here is a situation, and we go all the way around like that. This happens spontaneously in water. And what that does is it means that this part here interacts with the water fine because that's where the negative charges are. This part here interacts with the water fine. And these fatty acids, they're happy being together because what we talked about before is that nonpolar molecules tend to be attracted to other nonpolar molecules. One of the rules in chemistry is that like dissolves like. Okay? So if I took some oil and I melted some butter, that oil and butter would probably dissolve very well within each other, okay? If you have water and another polar molecule like alcohol, they dissolve very well in one another. So these two things are kind of like mingling with each other because they're attracted to one another. They are very similar molecules, so they can hang out together without repelling each other, okay? So now we've achieved a situation where we have this circle that acts as a barrier. So we have a water environment out here and a water environment in here. So you can see that this is very easy. It's easy for this to function as a protective outer layer for the cell. This up here is an example of something called a micelle. Micelles are circular membranes that are formed by phospholipids that only have one tail, okay? And you may have heard of micellar water if you're a person who's taken off makeup or used any kind of micellar wipes. And the reason that these work so well is because makeup is often, it has a lot of like waxes and things in it, but then you also want to use this part here that's water soluble to get off any of the water soluble part of your makeup. So micellar water works really, really well because it can get at the waxes in your makeup, as well as any of the other water-soluble stuff that you need to get rid of, okay? All right, so let's think about these phospholipid bilayers. So the remainder of this chapter is really gonna be honing in on these phospholipid bilayers that make up cell membranes, okay? So let's look at them a little more closely. So here I have a phospholipid bilayer. So here's our phosphate group here. Remember, that's negative, so it's dissolved very easily in water. And here's the other side, dissolved very easily in water. And this interior region, remember, this region here is nonpolar. Now, this is going to set up some interesting features. So what that means is things that interact with water, they can interact with these polar heads here, right? They can interact here and here. But if it's something that's dissolved in water, it's going to have a hard time crossing through this nonpolar region because if it's soluble in water, then it's not soluble in oil. Here's what I mean. Let's take a look at things that can cross lipid bilayers. Things that are really small, like oxygen, carbon dioxide, or nitrogen. These are small, non-polar molecules, and they have a pretty easy time getting across these lipid bilayers because they can fit very easily between these larger molecules. So they can get across pretty easily. They have what we call high permeability. Water, on the other hand, it's small, but it's charged. So it can sneak between these, but it has a hard time because it also struggles with this interior region here. So you have some water that can sneak through, but not all water can sneak through, okay? In order for water to sneak through, there has to be room in between these molecules so it can get from one side of the membrane to the other. And it's got to move quick because it does not like this nonpolar region here. Now let's look at something called glucose, right? We've talked about glucose before. It's got 6 carbons, yada, yada, yada. And it's got a double bond here and a hydrogen, and there's a whole bunch of oxygens over here. 1, 2, 3, here we go. OH, okay, you're with me. I'm not gonna draw out the whole molecule, but we've got a lot of oxygens in glucose. So glucose is polar. And because we have 6 carbons, it's bigger, okay? Polar and big. Glucose struggles. It cannot get across this membrane very well at all. It does not fit between these molecules. And because it's polar, it cannot manage this nonpolar interior of the lipid bilayer. And then the thing that absolutely cannot get across these membranes are anything with an actual charge on it, like a chloride ion, a potassium ion, or a sodium ion. These have minuses and pluses. These are absolutely repelled by the nonpolar interior of these phospholipid bilayers. So this sets up some interesting features for cells, right? We can get oxygen into our cells pretty easily. We can kind of get water into our cells. We might need some help. But if we want to get some of the things that we need for life, like glucose or sodium or potassium, if we want to get these things into cells, we're going to have to come up with a way to get them across these phospholipid bilayers to get them from the outside of a cell to the inside of a cell. All right? Now, one thing that I wanted to mention that I mentioned briefly earlier is that when we have lipid bilayers that are making up a cell membrane, sometimes you might want this to be a more fluid membrane, sometimes you might want it to be a more rigid membrane, and what determines the rigidity of cell membranes is the number of unsaturated bonds, okay? So here we have a phospholipid and we have an unsaturated bond. Here we have a bunch of saturated bonds. So this is going to be a more rigid cell membrane. This is going to be a more loose cell membrane, okay? But the thing that you need to know about cell membranes is that in general, you need to think of them as oil bubbles, and that these individual lipid molecules are in constant motion on the surface. So if you've ever blown a soap bubble and you can kind of see that the bubble it's got swirls on it where the bubble solution is actually kind of swirling around on the surface of that bubble, then that's a really good analogy for how a cell membrane works. And the phospholipids in this cell membrane are swirling around on the surface of that oil bubble. But the oil sometimes can be thicker if you have a lot of saturated hydrocarbons, or it can be runnier if you have a lot of unsaturated hydrocarbons. And I want you to think about, when would you want your cell membrane to have thicker consistency, and when would you want it to have thinner consistency, okay? All right, that's where I'm going to stop for this lecture, and we'll pick up with our next lecture talking about how we can transport things across these cell membranes. Bye-bye.